So let's now go over paper 4, which is the extended theory paper for biology. That's IGCSE from Cambridge International Examination, CIE, and that's a summer, May-June 2017, variant 1, so 0610/41 is a code. Let's get started.
Okay, Anna, on 3.
Okay.
1.
You tell me when. I'm ready to go.
2.
I was born ready, yes.
Calm down.
Question 1. Fat is a necessary component of the human diet. A. State 3 ways in which the human body uses fat. Okay, it is 1. The storage of energy. Okay, we use fat as a storage form of energy so we can use it for respiration. We also use it for insulation. Okay, it's insulation to reduce heat loss or as myelin on nerves. Those are actually the same marking point, so they're both insulation. So one of them is to insulate against heat and one of them is to insulate the nerves so that the electrical impulses don't travel to everywhere in the body, just along where it needs to go. And another one is if you have a layer of fat, it protects the body. So it's for protection against mechanical damage. It's a shock absorber. Okay. Other things you could say is that they're used to make some hormones, and they're used to make cell membranes, and we can absorb vitamins with fats, because some vitamins are fat soluble. All right. So the arrows in figure 1.1 show the pathway of fat in part of the alimentary canal. Okay, so it comes down through the esophagus, through the stomach, through the duodenum, down through the small intestine, goes to the small intestine and includes a few other organs on here, the pancreas, the liver, gallbladder, and then it goes throughout the rest of the body. Okay. B. State the name of 1. The enzyme secreted by the pancreas that digests fat. Okay, the enzyme that digests fat that's secreted by the pancreas is called lipase, because it digests lipids. 2. The products of chemical digestion of fat. Okay, that is fatty acids and glycerol. This says products has to have at least 2 things. Okay, you need both of them to get your 1 mark. Okay, 3. The liquid that is produced by the liver and stored by organ P in figure 1.1. Okay, the liquid that's produced by the liver and stored by organ P, well, that is the gallbladder. So the liquid is called bile. Bile. So organ P, as we just said, was the gallbladder. All right. C. Explain what happens to ingested fat at R in figure 1.1 before chemical digestion occurs. So the fat comes down through the esophagus into the stomach, into the duodenum, and then before it gets to the enzymes from the pancreas, what happens? Well, the main thing is the bile is secreted from the gallbladder. So the bile emulsifies fats, and that means it breaks the fats down into smaller globules. What this does is it increases the surface area to volume ratio, and that is one of those key words that comes up a lot in this topic and in a couple other topics. Okay, and the reason why it increases the surface area to volume ratio is because it allows the enzymes, the lipases, to chemically digest afterwards. So the complete answer, bile emulsifies fats. This means it breaks fats down into smaller globules. It increases the surface area to volume ratio for enzymes, lipase, to chemically digest afterwards, because this is talking about before chemical digestion occurs. That's 4 points. You probably don't need all of them. However, they're pretty much all important for discussing what happens with bile. D. Explain how the products of fat digestion are transported from Q to the rest of the body. Okay, so Q is the small intestine. So first of all, the glycerol and fatty acids are absorbed by microvilli, and they get absorbed into 2 places. They can enter the capillaries, or they can enter the lacteals. So it enters the capillaries to travel by blood, and it also enters the lacteals to travel in lymph via the lymph vessels. and the lymph empties into the blood. All right. One possible effect of too much fat in the diet is coronary heart disease. E. Describe how too much fat in the diet may cause coronary heart disease. Okay, coronary heart disease is caused when fat is deposited in walls of arteries. It's called coronary heart disease because the fat is specifically deposited on the coronary artery. Okay, so the fat is deposited in walls of arteries, and coronary heart disease has fat depositing on coronary arteries. So basically, fat deposits in any artery in the body, not veins or capillaries, it's the arteries. And the coronary heart disease is because it's specifically also in the coronary artery. Okay, this is called atherosclerosis, and those deposits can be called plaques or atheromas. All right, so those are other keywords that could get you some marks there as well. So what this does is the arteries get a very restricted amount of blood flow, and they may become blocked. So the arteries get restricted blood flow and may become blocked. And what this does is it means the heart muscles get less or no blood to them. And you have to be specific here that it's the heart muscle, not just the heart in general. It's the heart muscles that get less or no blood flow. And what this means is that there's fewer or no nutrients or oxygen reaching the heart muscles, which is very bad. So fewer or no nutrients and oxygen reaching the heart muscles. Okay, so these are a bunch of marks you can get. You can also get points for any other valid point, as it says. So this could be that the blocking of the arteries, the coronary artery, causes increased blood pressure. You can also say that it promotes blood clotting in other areas, or in the heart or in the brain, and that's called thrombosis. Okay, and when the heart muscle can't get oxygen, it can't respire aerobically, so it has to respire anaerobically. That produces lactic acid, and there's a whole bunch of other problems. So there's at least 3 marks you can get there. There's tons of marks you can get there. F. Describe and explain how coronary heart disease can be treated. So we need to describe it and explain how. Okay, this is a big question. This is worth 6 marks. All right. So there's a few ways that coronary heart disease can be treated. Basically, it can be treated with drugs, or it can be treated with surgery. That's really pretty much it. Okay, so coronary heart disease can be treated with drugs such as aspirin. And there's other drugs they can use, warfarin and a few other things. Okay? And the reason for this is it reduces the risk of blood clotting. So this reduces the risk of blood clotting. Okay? As we said, there's also the surgery method. And that's very invasive, but it works. Okay, so coronary bypass surgery attaches a piece of one blood vessel, and it attaches it around the blocked vessel so that blood can flow around the blockage. So coronary bypass surgery attaches a piece of blood vessel around the blockage so that blood can travel around the blocked artery. Okay, another thing they can do is they can put in a stent. So they can put in a stent, which is a tube to hold the artery open and stop it collapsing. They can also put in a balloon. That's called angioplasty. So angioplasty is they can put in a balloon and inflate it to widen the artery. Okay, the whole purpose of this is to restore the proper blood supply to the heart muscle so that it can respire and get nutrients. So this is to restore blood supply to the heart muscle. Okay, specifically to the heart muscle, again, as I said, not just the heart in general. Okay. Question 2. The genes for antibodies are only active in lymphocytes. Okay, A. Define the term gene. So a gene is a length of DNA that codes for proteins. Okay, so a length of DNA, specifically it's a full length of DNA that codes for proteins. It's a 2-mark question, so you have to say what the purpose is as well. B. Lymphocytes produce antibodies. Outline the role of antibodies in the defense of the body against pathogens. Okay, so antibodies, their purpose is they are able to locate specific antigens. Okay, so antibodies attach to antigens, and antigens are located on pathogens. So antigens are located on the pathogens, or located on the outside, and they are just markers that allow the antibodies to identify them. Antibodies are specific. One antibody is specific to one antigen. So each antibody is specific to one antigen because their shape is complementary to the antigen. Complementary is a good word there. They are not the same. So if you think about it, you have let's say you have a bacteria with one antigen, which this is a very strange antigen. Okay, so this is the shape of the antigen. The antibody basically has an inverted shape. It can't be exactly the same. It has to be exactly opposite. It's not the same as its opposite. It's complementary. So antibodies can do two things. They can either destroy the pathogens directly, or they can mark them for destruction by phagocytes. So antibodies destroy pathogens directly, or the antibodies mark pathogens for destruction by the phagocytes. All right. C, figure 2.1 is a drawing made from an electron micrograph of a lymphocyte that produces antibodies. Okay, lots of structures in here you should know. Table 2.1 contains statements about the structures visible in figure 2.1. Complete table 2.1 by naming the structure and identifying the letter that labels the structure. The first one has been done for you. Okay, so let's first of all name the structure, then we'll find the letter. Okay, so the structure that stores genetic information as DNA, that is called the nucleus. Okay, the structure that provides energy for making antibodies, that's the mitochondrion. The structure that is the site of production of antibodies, well, you could either say the ribosome, or you could say the endoplasmic reticulum. That's one or the other. Don't do both. They only ask for one. They're both correct because the ribosomes are on the endoplasmic reticula. But I would suggest don't write ER, because sometimes they don't like that. Sometimes they're okay with it. Sometimes they don't like that. So just write the entire thing if you're going to write endoplasmic reticulum. And the structure that is the transport of antibody molecules for release into the blood, that is called the vesicle. Okay, so let's look to see what we can find on the figure. Okay, so here are the structures. We have, first of all, the outside, we have the cell membrane. The second one, that's the nucleus. You can even see the little pores in it. C, that's pointing to that little dot, that's the ribosome. D, that's the cytoplasm. E, that's a mitochondrion. F, that's a vesicle. And G, that's the endoplasmic reticulum. All right, so let's fill in that table. So the nucleus was B. The mitochondrion was E. If you wrote ribosome, it would be C. If you wrote endoplasmic reticulum, that would be G. And a vesicle is F. But remember, only put one of these down. They don't want you to put two. And D, state the name of one type of cell other than a lymphocyte that is involved in the defense of the body against pathogens and describe its role. Okay, there's lots of different cells you could put down. One that you could do is a phagocyte. And the purpose of this is it ingests the pathogens to destroy them. Avoid words such as eats or attacks. It doesn't eat the cells or the pathogens in the same way that you eat a hamburger, and it doesn't just attack them. It doesn't use weapons or that sort of thing. It does ingest them with phagocytosis. All right. Other things you could do. You could say platelets release substances to promote blood clotting to stop pathogens from entering. You could say epithelial cells, so skin cells, provide a barrier to stop pathogens from entering. Goblet cells produce mucus to catch the pathogens. Ciliated epithelial cells, they move mucus, which also contains the pathogens, away from the gas exchange surfaces. Acid-secreting cells in the stomach make hydrochloric acid to kill bacteria. One thing you cannot put down, though, is white blood cells, because a lymphocyte is a type of white blood cell. It's not very specific. Question 3. Heroin is a drug that acts on the nervous system. A. Define the term drug. So a drug is any chemical at all that you take into your body that modifies any chemical reactions that take place. So a drug is any chemical substance taken into the body that modifies chemical reactions. All right. There are pain receptors in the skin. These receptors transmit impulses along sensory neurons to the spinal cord. Figure 3.1 shows the synapses between sensory neuron A and a relay neuron and sensory neuron B and a relay neuron in the spinal cord. Figure 3.1. Okay, this is figure 3.1. So this is sensory neuron A, this is the relay neuron, and this is sensory neuron B, and this is the relay neuron there. All right. Figure 3.2 is an enlarged view of the synapse between sensory neuron A and the relay neuron, as indicated by the circle on figure 3.1. Okay, so this is an enlarged view of a synapse. All right. B. Describe how impulses are transmitted across the synapse. So in order to transmit an impulse across the synapse, vesicles that contain neurotransmitters move towards the cell membrane. So these vesicles fuse with the cell membrane. So the vesicles containing neurotransmitters move towards the cell membrane, and the vesicles fuse with the cell membrane and release neurotransmitters. These neurotransmitters are released into the synapse, and then they diffuse across it. So neurotransmitters diffuse across the synapse. When they reach the other side, they bind to receptor proteins on the cell surface. So neurotransmitters bind to receptor proteins on the cell surface, and the reason they can do this is because the neurotransmitters and the receptors are complementary, and this results in an impulse being passed through the neuron. So the neurotransmitters and the receptors are complementary, and it results in an impulse being passed through the neuron. Good. C. Suggest how the structure of a synapse ensures that impulses travel in only one direction. Okay, so the reason why they can travel in only one direction is because the neurotransmitters are released on one side of the synapse and the receptors are located on the other side. So neurotransmitters are released on one side of the synapse and receptors are only located on the opposite side of the synapse. D. When an impulse arrives along sensory neuron B, a different neurotransmitter is released. This prevents the production of an impulse in the relay neuron. Molecules of heroin have a similar shape to the neurotransmitter released from these neurons. Explain how heroin affects the function of the synapse. Okay, so basically heroin is complementary to receptors for endorphin, and endorphins are the feel-good neurotransmitters, the neurotransmitters that tend to make you feel good. So heroin is complementary to endorphin receptors. So heroin, when it's taken into the blood, it diffuses into the synapse and binds to these receptors. So heroin diffuses into the synapse and binds to receptors for neurotransmitters. Okay, so when it binds to these receptors, it blocks the neurotransmitters. So that means they can't get any neurotransmitters entering the receptor. So the signal's stopped. So it blocks neurotransmitters from entering receptor site. Okay. E. List 3 stimuli other than pain which humans can detect. Okay, so humans can detect light. We can detect temperature. We can detect sound. And we can detect lots and lots of other things. It only says 3, but if you want something else, you can say chemicals. We can detect taste, smell, pH. We can detect pressure. You know, if you're touching something because you can detect the pressure on your skin. You can detect position, basically gravity. You can detect gravity. You can detect movement. You can detect if your muscles or tendons are stretching. So stretch. Okay, there's lots of things you can put down there, but only put down 3 because it asks for 3. Question 4. Figure 4.1 shows part of the circulatory system of a fish. The arrows show the direction of blood flow. All right, so we have blood flowing up here through the heart, coming up and going through the gills, then coming back down here to the body. All right. A. The circulatory system of fish is described as a single circulation. State what is meant by a single circulation. So, we have a double circulation because our blood goes through the heart, then it goes to the lungs, then it goes to the heart again, then it goes to the body. So in a fish, blood travels through the heart once in one cycle of the body. So blood travels through the heart once in one cycle of the body. That's what makes it single circulation. Okay, it goes through the heart, then it just goes to the body, then it goes to the heart. And the gills are on the way to the rest of the body. B. State the letter of the blood vessel in figure 4.1 that contains blood at the highest pressure, which means it doesn't want a name of it, it just wants the letter. Okay, blood is going to have the highest pressure when it's just left the heart, when it's just been freshly pumped. So D is going to have the highest blood pressure. So D. C. The gills are the site of gas exchange. State two features of gas exchange surfaces. Okay, so a good effective gas exchange surface should have a large surface area. They should have a large surface area, and they should also be very thin, generally 1 cell thick. So a thin surface, 1 cell thick. Other things that you could put down, you could say it has to be moist. So a good site of gas exchange would be a place where it's moist because the gases diffuse in the moisture. A good gas exchange surface should also have a good blood supply because you want the gases to go into the blood. So lots of capillaries around it. They should have a good amount of air or water around the surface to get oxygen. So in humans, it's obviously the lungs. We have the alveoli where we have lots of air going in. In the fish, they need the gills where they can have lots of water going past the gas exchange surface. And it should be permeable to gases. But remember, it's only asked for two features, so you only write down the two that you're most comfortable with. 5. The giant quiver tree, Aloe Palancy, shown in figure 5.1, is an endangered species. I know I've never seen it before. These long-lived trees grow in harsh environments. Some populations of A. Palancy are found within the Richtersveld National Park, but one population is found just outside on a mountain called Cornell's Kop in southern Africa. All right, pretty cool looking plants. A1, state the genus of the giant quiver tree. Okay, it says the giant quiver tree is called Aloe Palancy, which means that aloe is the genus and Palancy is the species. So it's aloe. And remember, genus and species when it's typed are always in italics. 2. Explain why the A. Palancy trees on Cornell's Kop represent a population. So these trees are an isolated group of individual plants of all the same species, very important, living in the same area at the same time. Okay, this is an important definition you need to know. So an isolated group of individual plants, or individual organisms, if they're talking about something else, of all the same species living in the same area at the same time. All right. B. Suggest 3 reasons why the giant quiver tree is an endangered species. Okay, it only wants 3. There's lots you can choose from, but the key word there is suggest. And suggest means it may not be true. You're just trying to think about what could be reasonable. Okay, there could be deforestation. So it could be cutting down trees. Okay, it could be because of climate change or global warming. Choose one. Don't write down both. Okay, that'd only be 1 mark. Okay. It could be because there's an increased number of new invasive grazers. Okay. So an increased number of new grazers. So grazers eat plants, basically. And the key there is they need to be new ones because there would already be some grazers there and the population would have been stable at some point, probably, with the number of grazers that were there. So it's why is it decreasing? Why is it an endangered species now, but it wasn't in the past? Okay, so these are all about changes. What has changed? Other things you could put down, you could say there was a change in the land use. Maybe the land that they've been growing on is now being used for a shopping mall. Maybe there's desertification, which basically is when you have deforestation and global warming, the area can turn into a desert. Maybe there's pollution. So maybe there's something contaminating the groundwater. Maybe there are plant hunters. So this is a very endangered, very interesting looking plant. So it could be that people are cutting down the trees because they want, or taking the trees home because they want a sample of that plant. There could be a new disease or a new pest attacking the tree. And there could be that it's not the tree that's getting attacked. It could be the pollinators that are dying away. So they might have a lack of pollinators. There's lots of things that you could put down. Only choose 3, but choose 3 you're most comfortable with. C. It was estimated in 2005 that the total number of giant quiver trees in the wild was less than 3,000. Sounds like a lot, but it's not, which is considered to be very low compared with other tree species. Explain the risks to a plant species of having very small numbers. Okay, so a plant species that has small numbers there's a high risk that they could go extinct. Okay, so that there is a high risk of extinction with low numbers. Okay, when there are fewer numbers, there's less chance that they could get pollinated, because pollination is generally by chance, just happens to happen. If it's spread by the wind or it's spread by insects going from plant to plant, it still is often luck. And the more plants there are, the more likely they'll be pollinated. When there are fewer plants, there's also less genetic variation, because any pollination that occurs is happening from a smaller gene pool. So there's less genetic variation. And because there's less genetic variation, there's also a high risk of genetic diseases, because they are more likely to occur in populations where there's less genetic variation. So there's a high risk of genetic diseases. This is because genetic diseases are often passed by the recessive allele, which means if there's less genetic variation, you're more likely to get 2 recessive genes in one organism. Okay? Also, if you have a very small population, they're more vulnerable to things like pests or diseases, or even a catastrophe. Maybe there's a landslide and it wipes out a few hundred trees. That's a big deal if you only have a few thousand. So small populations are more vulnerable to pests, diseases, catastrophes. So another catastrophe that could be a big problem to small populations, things like volcanoes. If this is on an island and half the island is destroyed by a volcano, that's a big difference in the population. Okay, so that is the end of this part. Let's go on to D. D. The population of A. Palensi trees on Cornell's Cup was surveyed and photographed at 4 sites, A to D, from 1937 onwards. Researchers took photographs at all 4 sites in 2004 and compared them with the original photographs. The results are shown in Table 5.1. Okay? So if you notice, these are at different sites. They're not the same site being sampled at different years. They're different sites being looked at at different points. 1. Calculate the percentage decrease in the number of living trees at Site B from 1953 to 2004. Okay, so we're looking at Site B. We're looking between 1953. So we're looking at how many trees there were in 1953 and how many trees there were in 2004. So the percent decrease equals the change in the number divided by the original number times 100, because it's a percent. All right, so that equals 9 to start with. There's 5 afterwards, divided by 9 times 100, and that equals 44%, 44.44444%. So 44%. All right. 2. Describe what the analysis of the photographs shows about the population of A. Palensi on Cornell's Cup. Okay, so if we look at this table, there's lots of things we can see. Okay, we can see that at every single site, the number of trees has gone down. None of them have gone up. All right, you can see that site D has the highest annual mortality rate. Okay, and site B has the lowest. However, site D has actually lost the fewest number of trees. It's just the percentage of deaths per year. Okay. You can also see that it's actually quite difficult to compare these numbers because they were taken at different sites at different times with a different number of starting trees. It's actually quite difficult. If you look at the original photographs, site A has the most trees from the original photograph, and site C has the least trees from the original photograph. You can also see that site A had the most number of trees and site C had the lowest number of trees. All right, let's put some of this down in writing. Okay, so it showed that there's a decrease in populations at all the sites. And you can say that site D has the highest mortality rate, or you could talk about the lowest mortality rate being site B. So it showed that there was a decrease in populations at all sites, and site D has the highest mortality rate. It also showed that site A has the most trees in the original photograph, or C has the least. So site A has the most trees in the original photograph. You could also get a point from just quoting some data points from the last column, from the mortality rate, and you have to include the units. Okay. Another thing you could say is that it's actually difficult to compare the changes over time because the numbers are for different sites. They're looking at completely different areas. So it is difficult to compare the changes over time as numbers are for different sites. So what this question asked was it will ask you to describe, you're just describing what you can see about that analysis. Describe the analysis. You're not explaining anything. You're just writing down what you see. Just looking at some trends. Which one's biggest, which one's smallest, that sort of thing. It's just asking whether do you understand what the table's showing, really. Question 6. Students investigated the effect of mineral ion deficiencies on the growth of radish plants. The seeds that were used in the experiment were from plants that had been self-pollinated for many generations and were therefore all genetically identical. Explain the advantage of using genetically identical radishes in this investigation. Well, the big advantage is that basically, if you see any variation at all, you know that it's because of the ions and not because of any sort of genetic differences. So any differences that are seen are due to non-genetic factors such as the amount of mineral ions available. So this makes it actually possible to make comparisons. You could also say that it improves the validity of the experiment, which is basically what the first two are saying. Okay. The radish seedlings were divided into 4 groups. Each group was grown in a different mineral ion solution as follows. Group 1 had the complete solution containing all the major mineral ions. Group 2 had a solution with all the major mineral ions except nitrate ions. Group 3 had a solution with all the major mineral ions except magnesium ions. And Group 4 had a solution with all the major mineral ions except phosphate ions. The apparatus used to investigate the growth of the plants is shown in Figure 6.1. This is Figure 6.1. Okay, so the solution came in, and these radishes are sitting inside the solution, and the solution came out. That means that there's going to be a good flow of ions. It's not going to deplete it from the soil or from the solution. It means that the mineral solution is going to be regularly replenished, so it's not going to use up what's there. Okay. B. State 3 other environmental factors that could affect the growth of the seedlings. Okay, there's lots of things that can affect the growth of plants. Okay, so humidity of the air is one. Okay, another one is temperature. Another one is the amount of light they're getting. Okay, that's 3 things, but there's lots of other things you could put down. As I said, it has 3 in bold, only put down 3. You could say the amount of CO2, amount of carbon dioxide, the pH of the solutions, the rate of the oxygen being supplied. So if you remember with the apparatus, it was the solution was put in and then taken out at the other side. So that would also contain oxygen in the solution. So the rate of the oxygen supply, the depth of the solution, the volume of the solution, or the density of plants. If you have 12 plants competing in one and 3 plants competing in the other, the one with 3 is going to have a lot more chance of getting good nutrients. Okay, so there's any of those you could put down, but just put down 3. So the results of the investigation are shown in table 6.1, and this is table 6.1. We have group 1, 2, 3, and 4. And that is the complete solution without nitrate ions, without magnesium ions, and without phosphate ions. They started with similar number of plants. Obviously, they probably tried to start with 10 for each of them, but this one, 2 died before, or didn't germinate. This one, 1 didn't germinate. This one, 1 didn't germinate. Okay, that happens. And what they did was they took the dry mass. The dry mass means they take out the plant at the end of the experiment, and they dry it out, and then they weigh it. So this is not including any water that's in the plants. And they have the leaves, the roots, the total dry mass, and then they took the average of one plant. Okay. C. Describe and explain the results for the radishes grown without nitrate solutions, which is group 2. Okay, so let's look at with the one without nitrate solutions. That's this group. Okay, so if you look at those, the mean dry mass is much smaller than the complete solution, the solution with all the mineral ions in. Okay, if you look at the leaf mass, it's smaller. If you look at the root mass, it's smaller, so it's smaller for both of them. Okay, another point will be for taking figures. So say the mean dry mass of a plant without nitrates was 216 milligrams, and the mean dry mass of a plant with nitrates is 374 milligrams. You're comparing using the table, so you should probably quote a data point. So plants without nitrate ions grew less than the control group, which was group 1. So the leaf and the root masses per plant are both less than in group 1. Okay, now you can quote a data point. So the mean dry mass of group 2 plants was 216 milligrams, and the mean dry mass of group 1 plants was 374 milligrams. Now is the explain part of the question. So why is this happening? So this is because nitrate ions are required to make amino acids for the proteins, and the proteins are used by the plant in many different ways, including for photosynthesis. And if you don't have as much photosynthesis, you don't have as much growth. All right. D. Describe the likely appearance of the radish plants grown in the solution without magnesium ions, which was group 3, and explain your answer. Okay, there's 2 things you could say. You could say the leaves would be yellow. The leaves would be yellow, and this is known as chlorosis. You could also say that the plants would be shorter and stunted. Okay, and that's because the leaves are yellow. Okay, and the reason why is because magnesium is essential for the functioning of chlorophyll, just like iron is essential for the functioning of our red blood cells to collect oxygen. So magnesium is needed for chlorophyll, and chlorophyll makes plants green. If you're saying the leaves are yellow, you should be talking about why they're yellow and not why their growth will be stunted. Okay, so yellow because the green isn't working, basically. Okay, if you mentioned that they would be shorter, you'd want to say the magnesium is needed for chlorophyll, and chlorophyll is needed to trap the sunlight for photosynthesis so plants can grow and get sugars for respiration. Okay, both of them would be fine, but the key there is magnesium is needed for chlorophyll. It's essential. E. Phosphate ions are a component of DNA. Suggest why the radish plants in group 4 grew less than the radish plants in the complete solution, group 1. Okay, so group 4 radishes have less DNA produced because phosphate ions are needed for DNA. So group 4 radishes had less DNA produced, and new DNA is needed for cells to divide by mitosis. That's because chromosomes are made of DNA. So new DNA is needed for cells to divide by mitosis, as chromosomes are made of DNA. So mitosis is very important because it's one way in which organisms grow. So mitosis is one way organisms grow. So DNA is also needed for protein synthesis. So DNA is also needed for protein synthesis, and proteins are needed for growth. Obviously, that's 4 marks for a 2-mark question. You don't need all of that. But as I said before, that gives you a little bit of wiggle room. Okay, we have reached the end of this paper. I do hope you found this very useful. If you have, please press the like button. And if you haven't subscribed already, please subscribe. We do appreciate it. And if you have any comments or questions, please ask them in the discussion section down below. We'd love to hear from you. And have a great day.